Protein synthesis begins in the nucleus of the cell where DNA is located and is the first step of gene expression. It begins when the enzyme polymerase unwinds and unstrips a part of the DNA, known as the promoter, in order to copy the genetic coding of the template strand. This becomes messenger RNA or mRNA. These regions have a start and stop signal, which identifies the code for the protein that needs to be created by showing what codons the mRNA needs to copy. The code of DNA is based on the 4 nitrogen bases of DNA: adenine, thymine, guanine, and cytosine, G pairing with C and T pairing with A. RNA polymerase binds nucleotides as it moves along the DNA with the complementary RNA bases, with the exception of thymine becoming uracil in the mRNA. This ultimately creates a new single-stranded mRNA which travels from the nucleus through the nuclear membrane to the cytoplasm, where it attaches to a ribosomal RNA. Once the mRNA has left the nucleus, the DNA returns itself to its original double helix structure and the mRNA is ready to begin translation. The mRNA provides the genetic replication to the cytoplasm, which allows for the beginning of translation. The mRNA attaches to a ribosome or rRNA, which reads the mRNA's genetic coding 3 nucleotides at a time, called codons. tRNA found in the cytoplasm contain a singular amino acid found on one end of the tRNA, and the anticodon found on the other. The complementary anticodon attaches to the codon of the mRNA, which provides the information that determines the amino acid. Once the codons are joined, a second tRNA will attach to the mRNA, carrying a second amino acid. These amino acids will bind together to form a peptide bond. Then the first tRNA and the anticodon will detach from the acid and mRNA, leaving just the peptide bond and the second tRNA. This process is repeated until the stop codon is reached. Then all of the tRNA will detach, leaving the newly formed peptide chain, and therefore ending the process. The mRNA dissolves and bases go back to cytoplasm. Once the peptide chain is formed, it then needs to go through protein folding to determine its biological function. This occurs in the cytoplasm, where the peptide chain begins to fold into a 3-dimensional structure, where it gains its shape and structure, which reveals its function. This process happens in 4 stages: primary, secondary, tertiary, and quaternary. It begins in the primary stage, which is simply the amino acids being held together by peptide bonds. The protein then begins to fold up in the secondary stage and can take either an alpha helix structure or a beta pleated sheet structure, both held together by hydrogen bonds. The tertiary stage is where the protein takes its precise structure in order to determine its function and is held together by a range of covalent and non-covalent bonds. The quaternary structure is when single peptides bond to other peptides to form a complete quaternary structure. Despite my model effectively displaying the process of polypeptide synthesis, it has some limitations. The amino acids are extremely simplified and the codons only have letters like G instead of guanine. Additionally, the codons are not cut to match each other like they should, and the nucleus is in an unconventional shape. I also only did 4 amino acids from a singular chain in my model, which is extremely oversimplified. Moreover, the drawing of the protein folding was messy and extremely broad by not going into detail about the insulin that my model was based on, and rather just explaining the process in general terms instead. This led to my explanation of the final structure and shape being extremely broad, which led to me not being able to determine the function of the protein once the folding was complete. My name is Josh Barratt, and thank you for listening to my model.